Hello everyone, my name is Ben and today I will be presenting my research project on identifying novel protein targets for the development of pan-influenza inhibitors, which I completed over the summer under the supervision of Dr. Matthew Shapira and alongside 3 other undergraduate research students. The influenza virus places a tremendous burden on our society. The World Health Organization estimates between 250,000 to 500,000 deaths associated with the virus every year around the world, with some studies even predicting this number to be much greater. We are obviously all aware of the global threat posed by coronaviruses, but some of the biggest global pandemics in our history were caused by influenza, including Spanish flu in 1918, Asian flu in 1957, and the swine flu in 2009, resulting in millions of deaths. This, of course, is in addition to the seasonal epidemics we experience every year with the seasonal flu. There are 4 types of influenza virus: type A, type B, type C, and type D. Types A and B are the only 2 types responsible for seasonal epidemics, as types C and D do not commonly infect human beings. Type A, though, is the most prevalent, the most mutable, in many cases can be the most pathogenic, and is the only influenza type capable of causing global pandemics. This is why our work currently only examines and analyzes influenza type A viruses. Within influenza type A, the virus can further be classified into different subtypes or strains based on 2 key surface antigens, hemagglutinin and neuraminidase, which I will touch more upon their functions a little later. There are 18 subtypes of hemagglutinin protein and 11 subtypes of neuraminidase protein, and it is the combination of subtypes that classifies each strain. For example, the strain H1N1 contains hemagglutinin of subtype 1 and neuraminidase of subtype 1. The strains most commonly circulating in seasonal flus are H1N1 and H3N2, and these subtypes are also often most frequently used for structural analysis. However, other subtypes like H2N2 or H5NY, where Y represents any neuraminidase subtype, are also high on the outbreak potential list for humans. For the development of a pan-influenza inhibitor, it should be able to account for a wide range of influenza A strains. The viral replication life cycle of the influenza virus is essential to the infection of host cells and proliferation of viral progeny. Once the virus has been transmitted to a new human host through aerosol droplets or direct contact, infection normally happens in the respiratory tract. Host cells have this surface receptor called sialic acid, and sialic acid is recognized by the surface antigen hemagglutinin on the virus. Upon hemagglutinin binding to sialic acid, the virus enters the host cell via receptor-mediated endocytosis, where the vesicle then fuses with the lysosome in the cytoplasm. Lysosomes have an acidic interior, and influenza takes advantage of this as part of its uncoating process. A channel protein called M2 pumps protons into the interior of the virus, which allows detachment of the viral RNA genome from other proteins in the virus. Fusion of the viral membrane with the vesicle membrane then permits the release of 8 viral RNA segments, along with associated viral proteins, into the cytoplasm, where they then translocate into the nucleus. Since the viral RNA segments are negative sense, they first need to be transcribed into positive sense RNA for replication. The viral RNA polymerase does this through a cap snatching process, in which it cleaves off the 5' caps of host pre-mRNA molecules to use as primers for transcription initiation. The viral RNA polymerase can now replicate viral RNA segments, some of which then take advantage of host ribosomes to produce more viral proteins. The newly synthesized viral RNA genome and associated proteins then bud from the cell membrane to form viral progeny. The viral progeny is finally released when the other surface protein, neuraminidase, cleaves sialic acid so hemagglutinin does not reattach, and the progeny virus is now free to exit and infect other cells. One of our best preventative measures against influenza virus, other than healthy physical practices like washing hands and covering up a sneeze, is receiving the seasonal flu vaccine. The flu vaccine varies in effectiveness from year to year. Since the influenza virus mutates very frequently through processes like antigenic shift and antigenic drift, it is generally recommended for individuals to get the flu shot every year. Some years, the vaccine matches up well with the circulating virus for that season, but other years the vaccine does not match as well, in which case the flu shot may actually provide little to no benefit or effectiveness. Additionally, compliance to receive the vaccine is relatively low. Only around 40% of Canadian adults consistently get the flu shot every year. Furthermore, just 44% of Canadian adults with chronic conditions get the flu shot, and these are high-risk individuals. The government actually has a goal of getting 80% vaccination rates in high-risk groups, but obviously 44% is well short of this target. Clearly, vaccines alone are not enough to protect us against influenza. The influenza A virus proteome contains 10 main proteins, all of which function to some degree in the viral replication cycle. Currently, though, only 2 of these proteins are targets for anti-influenza drugs approved for use in Canada, the M2 channel protein and neuraminidase. M2 inhibitors like amantadine and rimantadine function by occluding the channel so that protons cannot flow into the interior of the virus, disrupting the uncoding process. However, mutations that confer resistance to amantadine and rimantadine have become so widespread that M2 channel inhibitors are no longer even recommended as a treatment option for recently circulating strains of influenza. Now Canada relies on neuraminidase inhibitors like oseltamivir, zanimivir, and peramivir as the only approved antivirals against influenza, and this really represents the crux of the issue, the lack in diversity of available anti-influenza drugs. While neuraminidase inhibitors are largely effective pan-influenza inhibitors, there have been reported cases of observed resistance in the past, specifically a histidine to tyrosine mutation against oseltamivir and a glutamine to lysine mutation against zanimivir. Widespread resistance to these drugs, though unlikely, is definitely always a possibility, and given our heavy reliance on neuraminidase inhibitors, in addition to influenza virus's high mutation rate, there certainly exists the potential for future outbreaks. This really underscores the need for novel antivirals against influenza. This leads us nicely to our objectives and aims. The main objective of this research project is to examine the entire influenza A virus proteome in silico and search for any drug-binding pockets, and then assess whether any of these could become good candidates to target for a pan-influenza inhibitor. To ascertain the potential of the pocketome, it is important to assess how conserved the identified pockets are across different influenza A strains, as well as determine how druggable the pockets are if there is currently no known inhibitor. Following analysis of the influenza A pocketome, the next aim is to compare all pockets to determine which may be good targets and which may not be good targets. With the ultimate goal of developing a pan-influenza inhibitor in mind, pockets with good potential should be both druggable and well conserved across strains. The final aim is to present all of our findings through an interactive web portal for open access to the scientific community, like the Shapiro Group has previously accomplished with the SARS-CoV-2 pocketome. To meet these aims by the end of the project, we used the following approach to analyze the proteome. Each member selected one protein and performed a literature search to get a better understanding of the protein's structure, function, and if there are any inhibitors currently in development. Then we loaded all relevant structures from the protein databank into the protein ligand modeling software ICM Pro. ICM Pro has various functionalities which allow us to perform different types of analyses, visualize and represent our selected proteins, and create interactive documents with 3D slides, and the majority of the project was completed using this software. Some of the tasks we performed in ICM Pro included predicting binding pockets on the proteins, studying the effects of different mutations on ligand binding, and completing a multiple sequence alignment to determine percent conservation and identity of pockets. So let's go over my first selected protein, which is the polymerase acidic or PA protein. The PA protein, shown here in yellow, is one of 3 subunits in the viral RNA-dependent RNA polymerase complex, and the function of this complex is integral to the viral replication life cycle. As you may recall, this complex needs to hijack the 5' caps of host mRNA molecules in order to initiate viral transcription. The PA subunit is responsible for cleaving off the 5' caps, as it contains an endonuclease site in its N-terminal domain. A study has demonstrated that blocking PA protein's catalytic function by mutating evolutionarily conserved residues disrupts viral transcription. So, given its important role to the RNA polymerase complex, PA protein serves as an interesting target to explore. Taking a closer look at the endonuclease site, it cleaves 10 to 13 nucleotides downstream of the 5' cap. In the site, and necessary to the catalytic function, are two manganese ions serving as cofactors, which help coordinate the endogenous substrate of ribonucleoside 5' monophosphate for cleavage. Shown here is RUMP as an example. Now currently in the United States, there is an FDA-approved antiviral called baloxavir acid that occupies the catalytic site, which prevents endogenous substrates from binding and inhibits endonuclease activity. Baloxavir acid has a polar head group coordinating the metal cofactors and a lipophilic moiety making interactions with the rest of the active site through van der Waals forces. It is an effective and potent compound against PA protein's endonuclease activity, with a reported mean IC50 value of 2.5 nanomolars. However, there has been a mutation isolated from human samples where isoleucine at position 38 is substituted to a threonine, and this reduces susceptibility of PA protein to baloxavir acid, and this is due to the side chain of threonine being orientated differently so that van der Waals forces are disrupted. We can then use ICM-Pro to identify residues with side chains making interactions with baloxavir acid using the following command. Here are the interacting residues. To see how identical these residues are across influenza A strains, we performed a multiple sequence alignment. To accomplish this, we first downloaded sequences of our selected protein from the NCBI flu database, which in my case for PA, generated 28,000 sequences from a variety of subtypes. After aligning all sequences using an external tool called K-Align, loading the alignment into ICM-Pro, and linking the alignment to our reference PDB structure, we ran the following script in ICM. This created a table displaying the percent identity at each selected residue, as well as what other amino acids are present at each position. Here are the results of the multiple sequence alignment on residues interacting with baloxavir acid. Percent identity is high for all residues, but there are a few positions that are more variable. Positions 20, 24, and 37 have quite a few sequences differing from the wild type, so we want to investigate if this variability might have any impact on ligand binding, and we can do this by conducting a mutational analysis on ICM-Pro. We can use ICM-Pro to predict how a specific mutation at a position will affect ligand binding to the protein. The software calculates the change in binding energy of the ligand in kilocals per mole between wild-type and mutant forms of the protein. As the precision of this calculation is approximately 2.0 kilocals per mole, any mutations that produce changes in binding energy greater than 2 kilocals per mole are predicted to significantly impact ligand binding. Here are the results of the mutational analysis. At positions 20, 24, and 37, the amino acids threonine, histidine, and serine, respectively, were predominantly responsible for the variation. Given that the change in binding energy is less than 2 kilocals per mole for all three of these mutations, the variability observed from the multiple sequence alignment is not predicted to significantly affect baloxavir acid's ability to bind to the active site. This result supports the continued use of baloxavir acid as an anti-influenza drug. The fourth row of this table shows the predicted effect of the aforementioned isoleucine to threonine mutation at position 38 on baloxavir acid binding. In agreement with lab studies, ICM-Pro also predicts this mutation to have a significant impact on baloxavir acid binding, serving as a positive control for this analysis. There is research on other inhibitors of different chemical scaffolds that also target this endonuclease site, including 4-substituted 2,4-dioxybutanoic acids, phenyl-substituted 3-hydroxypyridinones, and 4,5-dihydroxypyrimidine 6 carboxylides. Generally, All of these inhibitors have these adjacent oxygen atoms to coordinate the manganese ions. Dihydroxybenzoic acid also has these adjacent oxygens and can bind the active site. However, it does not inhibit catalytic activity. This suggests that bulkier compounds making additional interactions within the active site are needed to actually inhibit endonuclease activity. I then used the ICM Pocket Finder tool to identify any potential ligand binding pockets in the N-terminal domain of PA. ICM Pro predicted 2 pockets, one of which encompasses the catalytic site and known inhibitors, and the other on another area of the protein. This non-catalytic pocket is rather shallow and serves no biological relevance according to the literature, so analysis proceeded only with the pocket identified in the catalytic site. Here are the residues comprising the pocket based on the following command from ICM Pro, as well as manually adjusting some of the residues. Here are the results from a multiple sequence alignment on residues comprising this catalytic pocket. This table also features percent conservation, which takes into account residues with similar physical properties. So for example, lysine at position 19 is slightly more conserved than it is identical, because arginine and lysine share similar physical properties, so they would be grouped together and considered to be conserved. Percent identity and percent conservation appear to be high at all positions, but only residues greater than 99.9% will be considered when calculating the overall percent identity and conservation of the entire pocket. While 99.9% is an arbitrary cutoff, it still provides some sort of metric that allows us to compare the conservation of all the binding pockets once the entire influenza A pocketome has been analyzed. To calculate the overall percent identity and conservation, it is just the number of residues greater than 99.9% divided by the total number of residues comprising the pocket. For this catalytic pocket in PA, the overall percent identity is 78% and the overall percent conservation is 91%. Because there are already known compounds binding and inhibiting this pocket, we already know that it is a druggable target and do not have to assess the druggability ourselves. PA protein also has a much larger C-terminal domain. The C-terminal domain of PA does not possess any catalytic function, but it does have an important role structurally for the polymerase complex. It creates a groove that allows the N-terminal tail of the PB1 subunit to fit through, and interfering with this protein-protein interaction at the interface could disrupt function of the entire complex. Currently, there are some classes of compounds that are predicted to inhibit this interface, but there are no structures solved in the protein databank of the C-terminal domain in complex with any small molecule inhibitor. After running ICM Pocket Finder, ICM Pro identified 2 pockets of interest that surrounded the groove for the N-terminal tail of PB1. These 2 pockets were combined, along with some additional residues, to form one pocket. Here are the residues comprising this pocket in the C-terminal domain of PA. A multiple sequence alignment shows that this pocket is also well conserved across influenza A strains, with an overall percent identity of 78% and an overall percent conservation of 89%. Because no known ligand currently occupies this pocket, we have to assess how druggable it may be. To do this, we used another software called sitemap. This program takes into account factors like size, hydrophobicity, and enclosure to generate an overall druggability score, predicting the pocket to be druggable, difficult to drug, or undruggable. Sitemap produces a D-score of 1.066, which predicts this pocket in the C-terminal domain to be a druggable target. To summarize the results on PA protein, there were 2 pockets of interest identified by ICM Pro, one in the N-terminal domain encompassing the catalytic site, and one in the C-terminal domain lining the interface between PA and another subunit. Both of these pockets are well conserved across influenza A strains, and both are druggable, with one already possessing known ligands and one predicted to be druggable by sitemap. This qualifies both pockets to be good candidates to target for pan-influenza inhibition. The next protein I selected to investigate is polymerase basic 2 protein, or PB2. This is another subunit in the RNA-dependent RNA polymerase complex, and it too plays an important role in the cap snatching process and thus the viral replication life cycle. So we learned that PA functions to cleave off the 5' cap, while PB2 functions to bind the cap. It recognizes and binds the methyl 7-guanine cap and positions it for cleavage by PA. Currently in the United States, there is a compound being researched called VX787 to inhibit PB2, which I will talk more about in a bit. So PB2 contains a cap binding domain, a mid-link domain, and a C-terminal domain. The cap binding and mid-link domains allow PB2 to adopt different conformations. The cleavage and priming conformations are important for the binding and hijacking of the 5' cap, but there's also this apo conformation that physiologically is unable to bind capped RNA. VX787 takes advantage of this by binding between the cap binding and mid-link domains to inhibit the protein. The drug not only physically blocks the methyl 7-guanine cap from binding to the cap binding domain, but it also stabilizes the apo conformation. There have been some mutations, though, that have been identified to confer resistance to VX787, such as an asparagine to threonine substitution that reduces affinity for the ligand and also destabilizes the apo configuration. ICM-Pro then identified which residues had side chains making interactions with VX787. Here are the selected residues. Here are the results of a multiple sequence alignment on residues interacting with VX787. From the NCBI food database, nearly 28,000 sequences were downloaded for PB2 protein. Percent identity is high for all residues, but positions 355 and 511 are more variable, so mutational analysis was conducted to see if the variability impacted VX787 binding. The third row in this table predicts the impact of the aforementioned asparagine to threonine mutation at position 510, serving as the positive control for this analysis. Isoleucine in place of valine at position 511 is not predicted to affect ligand binding, but lysine in place of arginine at position 355 does, as the change in binding energy is greater than 2 kilocals per mole. This is an interesting outcome, as lysine and arginine are conserved amino acids, but a substitution at this specific position likely alters the non-covalent interaction between ligand and protein. There were 340 sequences in the database with lysine at position 355, suggesting that they may be able to confer resistance to VX787. ICM-Pro identified a pocket encompassing VX787 between the cap binding and mid-link domains. Here are the residues comprising this pocket. The multiple sequence alignment shows that all positions appear to be well conserved except for position 271. The reference structure has a serine at this position, but the majority of sequences have an alanine residue. This residue does not interact with VX787, but alanine 271 has previously been found to bolster polymerase activity in mammalian hosts. This may be something to note down, but does not appear to be relevant in the context of drug binding to the pocket. Since this pocket is in fact already occupied by VX787, we already know it is a druggable target. The C-terminal domain of PB2 does not function in cap binding, but it is believed to play a role in host determination and pathogenicity of the virus. There is a highly basic surface on this domain which has previously been found to function in RNA binding. Within this basic surface is a key residue at position 627 that appears to be significant for host determination. Lysine 627 is almost exclusively found in human isolates, but glutamic acid 627 is commonly found in avian isolates. Glutamic acid 627 is found to decrease RNA binding activity of PB2 compared to lysine 627, which could explain why glutamic acid is not found at this position in human isolates. Researchers have proposed that targeting the surface of PB2 could be a potential mechanism for novel anti-influenza drug therapy. ICM-Pro did not predict any binding pockets directly on this basic surface, but it did identify one in close proximity. Here are the residues lining the small pocket. A multiple sequence alignment shows that this pocket is pretty well conserved, with an overall percent conservation of 80%. No ligands are known to occupy this pocket, so we must also ascertain its druggability ourselves. Sitemap produces a D-score of 0.776, bordering between difficult to drug and undruggable. Given how shallow the pocket was, in addition to the basicity of the surface, it is not surprising that this pocket is predicted to be less druggable than others. To summarize the results of PB2 protein, there were two pockets of interest identified by ICM-Pro. One between the cap binding and mid-length domains in the apo conformation, which encompasses VX787, and one in the C-terminal domain next to the highly basic surface. The first pocket is relatively well conserved, and it is known to be druggable, so it is a good candidate for pan-influenza inhibition. The second pocket, while well conserved, is not predicted to be druggable and may just be a dead end. As a group, we currently have 8 out of the 10 main influenza A proteins fully analyzed. These 8 proteins are PA, PB2, hemagglutinin, neuraminidase, M2, nucleoprotein, nuclear export protein, and non-structural protein 1. We can then compare the conservation of the pockets from all these proteins, which is shown here on this plot. On the x-axis is the overall percent identity, and on the y-axis is the overall percent conservation. Any pockets that have an asterisk next to them have been predicted to be either difficult to drug or undruggable by sitemap. Druggable pockets that are located closer to the top and to the right would make better candidates to target for pan-influenza inhibitors, because they are better conserved across strains. This includes pockets like the PA endonuclease pocket, the PA C-terminal domain pocket, the PB2 cap binding pocket, and the neuraminidase pocket. Pockets that are closer to the bottom and to the left on the plot are not well conserved and would make poor candidates for pan-influenza inhibitors, and these include the hemagglutinin pockets and a non-structural protein 1 pocket. Here are the major takeaways so far from this project, both from my individual work on specific proteins and from the collective group work on the pocketome. In PB2, the pocket encompassing VX787 is well conserved, supporting the current research that targeting this site could eventually lead to an improved antiviral. However, it is also important to be wary of possible resistance to VX787. The basic surface on the C-terminal domain has previously been suggested in theory as a possible mechanism for pan-influenza inhibition, but practically it is not a very druggable target and may be a dead end in terms of drug development. For the PA protein, the pocket encompassing the catalytic endonuclease site is very well conserved. While there already exists an FDA-approved drug targeting this site in baloxavir acid, the results provide further evidence that baloxavir acid is suitable as a pan-influenza inhibitor. For the pocket in the C-terminal domain of PA lining the interface, I believe this site holds the most potential moving forward for developing a novel anti-influenza drug. Not only is there a molecular basis for targeting this site, it is also very well conserved and predicted to be druggable. This pocket definitely warrants further exploration as a candidate for pan-influenza inhibition. For the entire pocketome, this project also gives us an idea of which proteins do not warrant further research for a universal inhibitor, namely hemagglutinin. While in an ideal world, we would wish to find multiple good candidates, it is just as important to uncover which pockets on which proteins make poor candidates and convey this information to the rest of the scientific community. This could save time and resources from being wasted towards research on otherwise unpromising targets. We met the majority of our aims set out at the beginning of the project. With the exception of two main proteins, we have searched the influenza A proteome for possible binding pockets. We successfully assessed the conservation and druggability of identified pockets, and then compared them to determine which may be the best candidates. While we have yet to set up the web portal containing all our findings, we have created interactive documents for each of the investigated proteins, which will eventually be used for the portal in the future. Next steps of this project are first and foremost to finish analysis on the remaining two proteins, which are PB1 and M1. Then we should set up the interactive web portal, which includes standardizing all of our 3D documents. Once we have the entire pocketome analyzed, a possible future direction this project could take could be to screen for and optimize ligands to bind the best candidate pockets using in silico methods. Furthermore, the approach we use for this project could also be applied to aid in the development of inhibitors against other viruses, like HIV, which can only provide beneficial and valuable information to the scientific community. That concludes our work on this project over this past summer. I would like to thank you for taking the time to listen, and I hope you're excited to explore the Influenza A pocketome once it is available.